The opener muscle is obtained from a crayfish walking leg, which is autotomized from the animal. Once the muscle is exposed, the innervation pattern can be revealed by various staining procedures. Synaptic responses in the form of short-term facilitation are readily recorded from the muscle. A focal macropatch electrode can be placed directly over identified varicosities along the nerve terminals. Synaptic responses are then recorded, which can be used for measuring synaptic efficacy at a quantal level from a defined location on the terminal.
Hi, my name is Ann Cooper from the lab of Dr. Robin Cooper in the Department of Biology at University of Kentucky.
Hi, I'm Robin Cooper. My student and I will show you a procedure to dissect the crayfish opener muscle and use it for recording synaptic responses with different stimulating paradigms.
We use these procedures in our laboratory to study synaptic efficacy and the relationship between synaptic ultrastructure and function. So let's get started.
Back in 1879, the zoologist T.H. Huxley wrote a book entitled The Crayfish, which is still regarded today as a comprehensive guide to the life history, anatomy, and physiology of this crustacean. Around this time in history, the innervation of the opener muscle in crayfish legs was being characterized, and physiological studies were already underway in muscles of the crayfish. In fact, the experiments in crayfish might possibly be the first to demonstrate facilitation at the neuromuscular junction, or NMJ. Dr. Richet published his primary research in his medical textbooks, which he used to teach medical students about muscle function. Over the next few decades, crayfish NMJs were being described anatomically and physiologically in respect to tension development and anatomy.